moving on. We broke up this first session just to give people a little bit of a break, but we saved the best for last. So we have Jen Wojcik, who's going to talk about current state of risk prediction literature.
Hi, everyone. Thanks for having me. I'd like to make it clear in the forefront here that I'm presenting the work of a lot of different people, predominantly the ClinGen Complex Disease Working Group, which Carlos is the founding chair of, as well as folks at NHGRI who helped organize this meeting. And so what I'm going to talk about today is ongoing work and sort of preliminary results that we have from this, looking at how we do PRS, how it's reported, and how we see it being published right now. So the ClinGen Complex Disease Working Group, the goal of this Complex Disease Working Group is to create a framework for evaluating and developing genetic risk scores. The way that we're going about this is, first of all, to do a literature review of polygenic risk scores, the way that they're published in literature now. While this, we can look through it through the lens of reporting, reflecting on prior statements for reporting, and seeing if we can expand them to meet the criteria that we need for today, as well as developing a best practices going off this knowledge base. After that, we can have an assessment of how we're doing, how the field is doing in terms of what they're publishing, what can we do better, and then to develop, again, at the end, an improved reporting guidelines and best practices. And this is an ongoing collaboration with NHGRI. So the preliminary literature review, we wanted to assess the current state of the field, and we started with 34 representative papers from the field. These are numerous different outcomes, including Alzheimer's disease, asthma, cancers, coronary artery disease, Parkinson's, and you can see them all there. And the following I'm going to describe to you based on very preliminary results of these 34 papers. We are looking to expand this and get more in depth, but this is just sort of what we have at this point. So for this talk, there's 3 areas of focus that I really want to hit on, and the first is to look at definitions. So I think it's really important going into this when we do reviews to actually define what we're looking at. Now this includes, right now, we see a lot of heterogeneity in what is a PRS, what goes into the PRS, how it's used, how you define the outcomes, and how you define the different subcomponents of a score model. And so, these often vary even when we're in the same outcome. And so this is really important for us to look at the literature and see how it's being defined. Next, we'll move into evaluation. So now that you have a model, how do you assess both its development, the methods and stats behind that, as well as how it performs, such as why were the certain SNPs selected and how are they weighted in the model? And then lastly, we want to do a review of what the impact of PRS is. So what do we see these going forward in the field, and what's the impact upon clinical care? So the first one is definitions, and this is really a very large topic and a really difficult one, I think, given how long, as was previously mentioned, the field has existed. So we found 3 main uses of polygenic risk scores. These include disease risk, so given you're a healthy individual or just non-diseased, what is the risk of developing an outcome? 2, we have disease and subtype diagnosis. So given you already have developed the outcome, how do we determine what the outcome is? And then lastly, disease prognosis. So now we know that you have a certain disease. How do we have an idea based on your risk score of how that disease is going to go? So the majority of papers that we looked at actually are disease risk. There are fewer of the other 2 subtypes. And this is really a top priority of the ClinGen Complex Disease Working Group, predominantly because this working group is coming out of ClinGen, which has had a lot of success with Mendelian disease, and looking at the risk of that sort of groups of disease. So, whoops. Okay, so next up we have these 3 different types and the way that you would use the risk score. There are different components that would go into this polygenic risk score. We're calling it integrated polygenic risk score. So, at the bottom here we have the genetics. This can be both a genome-wide, more agnostic based on a p-value threshold with LD pruning. And then there's also your more typical epidemiological variables. So demographics, age, sex, race, ethnicity, and then clinical information. And it's really important that we define all of these different variables very clearly in the risk scores, because as I said before, a lot of times you have heterogeneity in how the different terms are used, and when you go back into the methods, it's actually a very different definition of what you're looking at. So one of these examples that we come up against time and time again is the example of race, ethnicity, and ancestry. There is a lot of inconsistency in the literature of how we define this. This could be self-report. Do we look at genetic components, genetic ancestry? Do you rely on self-reported race, ethnicity if you know that it's a good surrogate, but it might not be completely accurate to what you're looking at? One paper mentioned here defines African American as an individual with over 50% African ancestry and a European American of over 90% European ancestry, but it's not clear where those thresholds would have come from, and we do see some heterogeneity in where those thresholds are for different papers. And this has really big effects on how you interpret those scores and the performance of those scores. And again, many of these studies are restricted only to those of European descent, which, as we've seen in the previous talk, can be problematic. And so, there's an additional component that I want to throw in here, which is that in the United States, a large proportion of the minority populations are admixed. And so how do we define and stratify these groups is really important as we move forward to make sure these groups get the translation to clinical care. And this is just throwing up the principal components analysis from Page, and I think this really hammers home the point. If we see here each dot is a person, they are colored by how they self-identify. These are predominantly US minority groups. And so we have Africa ancestry on the right-hand side here, East Asia on the top. The Americas goes back to the left, and then at the bottom here of Europe. And I think what's really important to note is that, got messed up, but they're not discrete. So there's no genetic way to really cut up these different groups to make a meaningful stratification for these minority groups. And I think that's really important when we go and look at these stratified scores, sort of how we define that and why we're stratifying is important. So, again, how would you stratify this? In Page, we didn't, but that's important to note. And again, I'm bringing up Alicia's figure from recent work here. This is important, and the definition is important because there is a lot of things that can happen based on how you define ancestry. And so, as you see here, there's reduced prediction accuracy given the non-European groups. Some papers have shown there's no effect at all based on the scores. And then there's also the flip-flopping that was previously described. And so I think that the take-home from here is that there's an inconsistency that it doesn't seem to be very predictable, and it depends on the trait, it depends on the epi of that population and that outcome. And so it's very important to keep all these things into consideration for every risk score to evaluate the influence of ancestry on that risk score. All right, so the next point is the evaluation. Now this goes to how do we adapt previously established evaluation criteria to assess model development and performance. So I wanted to make a note here that there has been a lot of work done for an analytical framework in previous years. And right here what I'm showing you is the EGAPP analytical framework. This is evaluation of genomic applications and practice and prevention. And these are sort of the key questions that need to be answered when it comes to evaluating these measures. And so this includes, is it useful to your population? What's known about the analytical validity, the clinical validity, and then what are the potential issues and potential harms and issues in this population? So these are very overarching questions that have been set up already for us to work within. Then the next statement we have here is the GRIP statement, recommendations for reporting. Now this was specifically for genetic risk scores. And so what you see here are these sort of checklists of what needs to be described in every single paper to be able to evaluate sort of how good or how useful this risk score is. Now this includes your very sort of standard study design of participants to be able to describe the people that go into creating the score and validating the score, as well as very clear statistics and your methods for that going in. So these are sort of all the different points that you want to make. And then for assessment for the results, we have here model fit, predictive ability, and other performance measures, as well as validation of the risk model. So these different frameworks and statements already set us up to the things that we know we need to hit. And now, since these are 2009 and 2011, how can we update and expand these frameworks to encompass the way that PRS are calculated now and published now, and what we hope to do with them in the clinic. And as I said before, there's a lot of literature already touching on this, and so we are currently going through a lot of this to create the best practices. And I think it's important as well to draw from guidelines that aren't specifically for genetic risk models, but as was mentioned this morning, other risk prediction models that have a lot of the similar, obviously the same characteristics. And again, there has been a lot of work specifically on polygenic risk scores as well that we're drawing upon to build these best practices. Through the review, we came upon this paper, and these are also helping us inform the different check marks of what we need to see in a paper for reporting. And it really is, when we think of a polygenic risk score, we need to think about how we describe the different components that would go into these. And the 5 factors that were previously established by Chatterjee et al. In 2013, we need to know what we need to estimate what the number of true risk SNPs compared to the total number included in the model, what the true effect sizes would be for these risk SNPs, the chosen significance level, the power actually of the underlying association test to reach that significance level. So this goes for both, more of a candidate study as well as the GWAS that a lot of these scores are built upon. And then again, the expected value of the estimated regression coefficients for the selected SNPs. So these are all information that we're drawing upon to create a best practices and the reporting framework. Okay, so the preliminary conclusions from this really cursory sort of first round of reviews is that we've seen that manuscripts vary widely in the reported measures. Validation practices are very inconsistent. Some papers don't have any validation, whether it be internal or external. Some have internal, and some have both internal and external validation. Now this is sort of, I think, what we would expect, but it's sort of concerning to know that there are people who are reading these papers might not be able to assess their validity, obviously, based on what's reported. So having some sort of framework and best practices say you really should do some sort of validation so that you don't look like you're overfitting your risk score. And then, we hope to have a full round of reviews completed shortly and expanding this review to get a better view of the field with a timeline of this summer. So we're hoping. So lastly is impact, so doing a review of the papers and sort of seeing what the possible impact could be, which would in turn sort of inform us to what the best practice would be, to know sort of where you want them to translate to. So these are 2 examples that the group highlighted with the review so far, and the first one is the coronary artery disease. This is the paper that was previously mentioned this morning. So there was a GRS constructed from 3 different published PRS. It was evaluated independently and integrated with a traditional CAD risk for clinical covariates. And so what they show here is that if you segregate the groups into risk based on quintiles for both the genetics and the clinical covariates, you do better than only genetics or only risk factors, which I think we all sort of knew, but it's good to see the evidence there that if you combine these things, you do better. And then the last one, the next one is with breast cancer in 2017. And what you see here is that there is a previous risk sort of algorithm based on family history and other components that was being used with breast and ovarian analysis. They combined 24 known breast cancer risk variants into the score. And so, 14% of the women they looked at would have reached that 20% lifetime risk with just the family history and the estimation from before. But for PRS would increase that number to 23%. So you capture more women who are at risk. And so this sort of highlights the different impacts we could see having, and therefore the kind of components that would be really important to have in a reporting framework to inform future translation. Okay. And so, I think as we all sort of agree, I think at this point that this remains to be tested, how we use this risk score in the clinical practice. And as the working group believes, that previously stated, that this requires adoptions of common standards across the studies. And there really needs to be a standard both on how you conduct a polygenic risk score with the proper measures, very similar to what we have for GWAS, which is very, a rigid sort of check mark of what you need to accomplish in your study, as well as reporting to clearly define the outcomes and phenotypes. Okay, so again, this is ongoing work. The results are expected this summer for publication. looking at the definition, evaluation, and impact of these different scores. So again, this is the work of a large number of people. At ASGRI, I just want to say that Rob, Catherine, and Cecilia have done a large amount of the work, especially getting these papers together and doing the first run analysis of this. I'm speaking on behalf of ClinGen, the working group, which you can see here, as well as help we have at Stanford to review the papers. And I have one more slide, but I just want to let anybody know that if you're interested in this work or would like to join the working group or have any thoughts on this, feel free to contact Hannah or I, and we can sort of recruit you into the working group or take your feedback into consideration. And so I have a few minutes left, and what I really wanted to do was we have questions for you. And so sort of what are the concerns that the group has for PRS reporting? What concerns do you have with the papers being published or not being published? Where are the gaps, and sort of where do you think the framework from 8 or 9 years ago needs to be expanded or updated to accommodate the way that the field is right now? Yes.
Thank you for that nice summary. I just wanted you to comment on, so you've looked at the literature, and it may be also useful to actually look at what genetic testing laboratories are doing. Because I think sometimes the research is a little bit behind in terms of already implementation in the clinic. The questions that we're asking in the clinic about PRS goes beyond the methodology. It's doctors are actually receiving this information now and wanting to act on it. And the question is, what should they do? And so I think the reporting for publication in the research literature should also at least go hand in hand with what's already happening in practice. And so maybe you could include a discussion about what laboratories are doing.
That's a great point.
Breast cancer
Okay, thank you.
Okay, before we do questions, though, Jen actually had questions for us. So would anyone like to answer her questions, or Jen, would you prefer we put these into the general discussion?
We can put these in general discussion. That's
Yeah,
fine
because
with me.
I think people are just dying to ask their questions and not your questions. So, but Mark said his is directly related. We will vote and see if it's right.
That's right. Yes, and then I will cast the deciding ballot. So ClinGen has done an excellent job in the Mendelian disease of, and ACMG for that matter, of defining criteria for gene disease association, developing some frameworks by which those are applied, so that we're beginning to have some rationality applied to what should really be considered a valid gene disease association versus what is, evidentially lacking. So the question that I have that I think directly relates to the questions that you're asking are, would such a process be applicable for polygenic risk scores? And it would impact both, because you would need to say, what are the necessary elements of a polygenic risk score report that would map, say, to the Richards criteria for variant interpretation? But then secondly, what is the threshold of evidence needed to say that this is a true association with clinical relevance? And so I'm just curious as to whether there's been discussions about using that type of a framework and adapting it for polygenic risk scores.
Yeah, that's exactly what we're thinking from ClinGen. So sort of the same idea of ClinGen, where they've done sort of on a disease basis, disease-specific recommendations to look at these different score, risk and identifying loci. We're thinking of having a sort of a very similar framework for this, where again it would be disease-specific because there are different sort of risk profiles and different needs of these different fields. We are thinking right now of starting first with sort of cardiac outcomes, as well as potentially breast cancer or cancer outcomes, as sort of the two, I think, most prevalent outcomes and risk profiles. But yeah, I think that's definitely where we're coming from and would be the goal of the working group.
Great. We had a question from Eric and one from Greg, and then we'll go into general discussion.
First, it was a great talk. We didn't get to clap because we didn't know you were done. Thank you. So my question is about these questions, really. can we answer the questions if we don't really know how the score is going to be used? So for example, sometimes it's used to put people into buckets, let's say for a clinical trial. The comparison with ClinGen, maybe it's not fair because there you're making it, you're trying to develop a diagnostic for a particular individual. And I'm not sure, I don't know the answer to my own question, by the way, but I'm not sure there's a one-size-fits-all here in terms of the criteria until we have just, the field has just a little more maturity of how we can use these risk scores to sort of improve the healthcare of the human condition, whatever.
Yes, I think there's sort of two levels of reporting that we sort of touched upon, and then the first level I think is really just intrinsic to good population research, which is that a lot of studies that come out are lacking in details of who the participants are, what the different covariates are, and for that I think it's good to have that's sort of a one-size-fits-all, that you need to have some sort of reporting as to who, for epi papers, your typical table 1 of who everybody is and how you defined outcome. But I agree that there is sort of this mismatch between the way the field is now and then versus Mendelian disease. And I think that will, take a lot of effort with the disease-specific sort of aspects of the different working groups in ClinGen and externally, to see if they're, what these different outcomes have in common that we can make sort of this universal recommendation versus what are really disease-specific or specific to a certain outcome in a certain setting. I think differentiating between those is really important, and that we're looking to that.
Great. And then Greg, if you could ask a quick question, that'd be great.
I guess my comment was on the concerns about PRS reporting, and it's just that the difference between analytical and clinical assessment is so important. So if you take the Inouye paper, for example, the meta PRS, yes, it performs better from the area under the curve perspective. It increases it by 1 or 2%. But if you actually take the 3 underlying measures plus the meta GRS and you ask on an individual level, the variability is massive. So somebody can get a score that says that they're in the 10th percentile or the 50th percentile for those 4 contributing scores. And the overlap between the people in the top 2 or 3% is actually quite low. So the assumption that a score that performs better in terms of sensitivity and specificity, then translating to being better in precision, is completely wrong. And so that's a concern, and I think a very important point that we need to keep in mind.
Okay, thanks.
Great. All right, thanks. So Jen will let you sit down, and then we'll go on to a more general discussion, which certainly can touch on topics of Jen's talk. And Peter, you may have one, so go ahead.